Hello.
Hello.
7 o'clock. Hello. I welcome you, everybody, to a new session of the SICAM Conference 2021. My name is Gabrielle Solland. I will be one of your hosts for this evening. This evening, we will be hearing about the proteomic differences of winter and summer bees, a presentation by Rachel Ward. Very welcome, Rachel. Rachel is in her third year PhD at the Maynooth University in Kildare in Ireland. And I also welcome very warmly Professor Kevin Kavanagh, who's her supervisor and has agreed to host tonight with me for the Q&A. And I'm looking forward very much to an interesting talk and an interesting Q&A. So, hello, Kevin. I will ask you to introduce yourself a little bit to the audience, who you are and what your background is.
Okay. Thank you very much, Gabrielle. It is a great pleasure to be joining you this evening to give you an insight into our work. And we're based in Maynooth University, which is in the town of Maynooth, Ireland, and we're located approximately 25 kilometers west of Dublin City. The university is the smallest university in the Republic of Ireland. We have approximately 14,500 students, and although we're the smallest, we consider ourselves to be the best. So, my research interests are in the area of microbiology, but also in how pathogens interact with host, whether it's a human host or an insect host. And by studying these interactions, we can uncover new ways to treat pathogens and to help the host defend themselves. I've had a long-term interest in insect immunology, and over 25 years ago, began looking at the immune response of Gileria to pathogens. And we were able to show there were strong similarities between how Gileria responded to pathogens and how humans respond. So we could develop a model system. This interest in insect immunology led me to an interest in the diseases of honeybees. And I've had a number of projects in this area where we apply different techniques to uncover how honeybees respond to pathogens or to various environmental stresses. Rachel's project has been funded by Clarins for the last 3 years. And in her project, we're applying a particular technique known as proteomics to uncover how bees transition from one stage to the other in their life, how they respond to pathogens, and how they respond to various treatments that they might be exposed to. So I suppose the question might be is, what is proteomics and why do we use proteomics? Well, we all know that the genetic information of a cell or an organism is contained on the DNA. And this contains all the genes required for the function of the organism. But even though a gene may be expressed, it may never end up coding for and producing a protein. So, for example, we can think of an architect drawing the plans for a house. And the architect might have specific features in the house, but in the building stage, these might become modified. So therefore, the plan may not be fully represented by the completed product. The same happens in cells. Many genes are expressed, but never end up producing proteins. So therefore, if we want to uncover what's going on in a cell, what's going on in an organism, it's better to look at the protein level. And this is what the technique of proteomics allows us to do. In her work, Rachel has applied this technique to uncover specific features in the life cycle of the bee and also how bees respond to pathogens and stresses. And this information will give us new insights into developing new and more effective therapies for controlling diseases of bees. So it gives me great pleasure to introduce to you Rachel Ward, a third-year PhD student, and I'm glad to say also a beekeeper. Rachel.
Thanks, Kevin, and thank you, Gabrielle, as well. So hi, everybody. Yep, I am a final year PhD student, well, my third year for my PhD, and I have been a beekeeper for the last 2 years now. And it has been a wonderful amalgamation of both being a beekeeper and studying bees. I thoroughly enjoy it and find the two go hand in hand quite nicely. And what I'll do now, if it's okay, is I will share my screen and we'll start the presentation on some of my work for you. So today I'm going to be talking about, like Kevin mentioned, proteomic comparisons, so using proteomic techniques to compare two types of honeybee workers, meaning they're going to be looking at the winter honeybees and then the summer honeybees, with sole focus on the workers. And it's a collaborative project. Oh, no, it's a collaborative project. Perfect. Between Maynooth University and I'm not sure if that's going to, hope you can see, a collaborative project between Clarence, Ireland and Maynooth University. So there are several aspects to my research, and I'm going to be talking about three in particular today. Excuse me. The first is looking at the development of honeybee workers throughout their life cycle. So I am looking to investigate how workers, through the process of age polyethysm, what they will adapt with their proteins in order to be able to fulfill the roles that they have to do within the hive, and then to be able to succeed as well. So, be efficient nurse bees, good builders, and then foragers. So that's one of the aspects I'm looking at. With that, then, that goes kind of quite parallel or hand in hand. I am looking at the time frame of which winter bees will emerge within the hive. We want to be able to have a look at, is there a window of opportunity, or is there a window in particular that we see a lot of winter bees emerging within our hives? And the reason we think this is important to investigate is because when you are, as a beekeeper, preparing your hives for winter, you're going to need to do a lot of monitoring, possibly apply some treatment to be able to ensure that your winter colonies survive through the winter to the following spring. So we think it's really important to try and identify a really good time frame as when these winter bees emerge, in order to be able to better protect them, understand their immune system, and hopefully increase the amount of hives that are making it through the winter. And the third aspect of this project, and the main purpose of my talk here today, is to investigate the differences between a winter worker and a summer worker. And there's a photograph of me there, beekeeping at the hives down in Teagasc. And here are our hives. They're based in Oak Park Institute in Teagasc, down in County Carlow. And what we did, myself, Mary and Danny selected 3 hives that we were going to do the full age comparison. We sampled these hives from March 2019 through to March 2020. And we did this using a method of queen caging and marking, which I will discuss now in a second. And just to give an insight as well as what Kevin said as to why we study proteomics, as everybody knows, you have DNA in your cells, which is transcribed into RNA and then translated into proteins. And this is a slide I use a lot with beekeepers to try and explain what proteomics is. We have Tom Hanks here on the bottom left-hand side of the screen, and Tom Hanks's DNA is the same in both, but the phenotype of Tom Hanks changes in the 2 photographs. And this is due to protein expression. Unfortunately, when you're looking at bees and the differences between the ages, you can't really tell what's a winter bee and a summer bee just by looking at the phenotype and just by looking at the bees. So we're using proteomics to gain a really good insight into what's happening in the bees in a real-time situation in regards to age, environmental stimuli such as seasonal changes, or the presence of a pathogen or treatment. So how we progressed with our study was that in the 3 hives that we selected, we caged a queen on a frame and left that queen caged for 2 days so that she laid up the whole frame. That cage was then removed, the frame and the queen were released back into their hive. And We left the frame within the hive for 17 days, allowing for the eggs to develop into larvae and for the cells to be capped. After which then the frames were placed in an incubator, and we waited for all of the bees to emerge from the frames. Once this happened, we gathered up the bees from each frame into individual basins, the one that's photographed here on the left, and we marked them on the top of the thorax with a bee marker. What we did then is that we sampled those bees we took a sample of those bees for emerged bees, and then we released them back into the hives. And then, as seen in the arrows there, we sampled the bees at 3 different time points that we were investigating through an earlier study that we have done. And we sampled the bees at an early hive stage, which would be the nurse stage, which was 10 days, at a late hive stage, which would be a builder or a guard bee, at 17 days, and then as a forager at 27 days. So we had emerged, early hive, late hive, and forager, and these were the samples that we used for proteomic techniques. The proteomic analysis was completed over a 3-day period using various steps. And for our winter versus summer analysis, I used a head, an abdomen, and a venom sac as our anatomical samples. But for our H. polythism study, I used just the head samples. So with our samples, I completed some dissection work, and with the abdomen, I removed the honey stomach and the integument. So we were just left with the exoskeleton and the fat body that lines the inner part of that exoskeleton. The samples were then placed in a lysis buffer, and all the cells were lysed and protein was released. This was then placed in acetone overnight to precipitate the proteins, which was then resuspended, digested overnight with trypsin, and then did a protein cleanup protocol using C18 columns before loading them onto the mass spec. From the mass spec, then the data was taken and has been analysed using several bioinformatics software programs. The principal one that I've been using is Perseus. One of the results then, or one of the really interesting parts that I found, and this is in my first year of beekeeping as well, so I was very intrigued with this, was being able to see the workers that we had marked in September, early and late September, and being able to photograph them in the following February. I just thought it was really amazing that even though I read it in the books that the winter bees live throughout the winter period, it was really nice to be able to see that this is in fact the case. So the bee on the left that is marked in green, indicated by the yellow arrow, was caged, the queen was caged for that bee on the 2nd of September. And then on the right-hand side, I can do that, on the right-hand side there, the 2 bees that are marked with white along the top with the 2 yellow arrows, that queen was caged on the 23rd of September. So approximately 5 months, those bees have been within the hive, and it was wonderful to be able to see them. So onto the proteomic side of my work then. These 4 graphs you see in front of you are called principal component analysis graphs. And what they do is they will read the samples being the bees that have been used. They're represented on these graphs as the hexagons, the squares, the rectangles, all in the various colors. So each of those is a sample. And the principal component analysis graphs will read all the proteins associated with those samples. And usually it is approximately 2,000 proteins for all of the samples that we're dealing with. This graph will cluster the samples based on similarity and separate them based on dissimilarity. And it's reading the proteins in their abundances and what proteins are present within our samples. What we can see if we look to the July part, I'm not sure if I have my little laser on. I can try and do that now. Pointer options, laser. So what we can see with our emerged samples is that in the July, they're completely separated from our early and our late hive bees, and as well, they're separated from our foragers. We can see here that there are 3 distinct clusters: emerged, early and late hive bees, and foragers. And this is to be expected, as at this time of the year, the early and the late hive bees will have very similar functions and roles within the hives. And the foragers will be extremely busy because of the time of year that it is. There will be a lot of foraging, and there will be a lot of plants and flora that they will be able to go and visit. So it is not surprising that there's distinction between the foragers and our hive bees. However, as we progress to August and September, we start to see a shift in the positioning of our samples within our PCAs, our principal component analysis graphs. We see in August that the late hive bees start to move and start to shift towards the forager proteomes and become a little bit more distinct from our early hive bees. So this is suggesting that maybe the lates and the foragers are actually more similar, whether it's the time of the year that a lot of the late bees are going out to try and get the last of the forage, or that the foragers are actually remaining within the hive. So this is something we want to do a bit more work on in terms of identifying what the weather was like and the flora availability here. But what's really clear is the distinction between the emerged bees with our early and late hives and our foragers, which is continued into September. And again, this pattern of our lates and foragers are clustered more closely together, with our early hive bees also being, positioned quite close to our late hive and forager samples, with once again our emerged bees being completely distinct. And what really becomes apparent in October is that our early, lates and forager samples are all clustered together, even though they are in a sequence, with the earlies being in blue, the lates in yellow and the foragers in green. What we see is, even with this sequential patterning of our samples, that they are still clustered together, indicating quite a high degree of similarity between our samples, and that there is still quite a large difference between our emerged bees to our early and late hive bees and our foragers. So what we wanted to do is investigate this further to see how the emerged bees might be related to each other, as with the early, the lates and the foragers. And to do this, we used several different graphs within Perseus. I'm showing you the final product, which is called a hierarchical clustering graph. What this is displaying is all ANOVA significant proteins, which were calculated based on protein intensities within the samples. And it's also including statistically significant differentially abundant proteins, which are calculated using volcano plots. This is where you do direct comparisons of samples. For example, October emerged and September emerged will be plotted together, and based on the intensities of the proteins present and the abundances of which they are expressed with the samples relative to one another, will be determined as statistically significant, and only those proteins are used within this graph. The very first message that we can take home from this graph is that our July and August emerged samples are completely distinct from our September and October emerged samples. And you can see this because of the positioning on the graph and the fact that there is quite a large degree of separation with the clustering on our sample analysis here. And what happens is, on this graph, where samples are indicated in this greeny blue color are down-regulated, and those in this orange color are up-regulated. So what we see is, even though they do share some patches of blue and some patches of orange, that they have been clustered separately from each other, indicating a degree of separation, and therefore specific proteins are upregulated in October and September that are not observed within our July and August samples. So this was extremely interesting to see that our September and October emerged bees seem to be quite distinct from our August and July bees. And the second really interesting aspect of this graph that we've noticed is that our October hives and foragers, so our early, lates and our forager bees, have a cluster that is upregulated that is not observed within our September or August or our July bees. And this cluster here, identified in this yellow rectangle, we have identified these proteins to be involved in xenobiotic metabolism, specifically cytochrome P450 proteins, and oxidative phosphorylation proteins, specifically cytochrome C oxidase proteins. So both proteins have been known to have a role with reducing oxidative stress, or with reducing ROSs within the cells and increasing longevity. So at the moment, we're really intrigued to see what else is happening within our September and October samples when they're emerged. And also, what is it with our October hive and forager samples that is causing them to be so distinct from other samples? And could this be the beginning of the proteomic fingerprint identifying the emergence of these winter workers? So following on from that, what we decided to do was sample bees in December, and these were taken at the door of the hive, and compare them to bees that were sampled in June. Again, these are all the same hives that were sampled at the late hive stage from the tops of the frames. And what we did is we did direct comparisons with our samples with the heads, the abdomens, and the venom sacs. what we can see again with these principal component analysis graphs is distinct, like highly distinct separation between our winter samples and our summer samples. And the really interesting thing about these three graphs here is that the overall percent separation, or the overall clustering, the overall separation between our PCAs with the samples equates to between 92.2 and 92.7% degree of separation. So it shows there's highly significant changes happening between these samples because they're clustered so far away from each other. And when we dive into this a little bit further and look at the specific protein changes that are occurring, we see that in this volcano plot, the summer proteins are identified on the left and the winter proteins are identified on the right. And we see that all of these proteins here in orange I've labeled as significant, and these in blue are significant for our winter bees. We see that the summer samples have a huge abundance of proteins found in the summer on the left-hand side here, which can be identified because of the large fold change that we're seeing. So this is the fold change is on the x-axis. That is not observed in the winter samples. So we see that the summer bees seem to have a much greater fold change of proteins, a relative fold change, meaning that there is a much higher abundance of these proteins identified in the summer that is not observed within the winter. And two particular groups in the summer that we've identified as highly relevant is quite a large increase in cuticular proteins within our summer sample, with a relative fold change ranging between 2.2 to 4,836, which is a huge increase in cuticular proteins within our summer samples, indicating that these cuticular proteins are expressed at extremely low levels within our winter bees. Another group of proteins we've identified as significant within our summer samples is the major royal jelly proteins, which again have a very large relative fold change abundance in the summer that is quite low in the winter in comparison, because all of these protein changes that you see here are relative to one another. Something that's very intriguing that we see in the winter bees is the increase in the xenobiotic metabolism proteins, with a fold change range of 6.62 to 92.35, meaning that these xenobiotic metabolism proteins, specifically cytochrome P450s, are observed at a much higher abundance in our winter bees, and therefore at a much lower abundance in our summer samples. So that's the specific protein changes that we see. But what we like to do as well is have a look at the kind of broader picture as to what's actually happening within our samples and how these proteins are related to each other. So what we do is we gather up the protein data and we run it through a BLAST koala search, which will use gene ontology pathways to see what groups or what pathways the proteins will fall into. And what we see here, as a very quick oversight, is that the summer workers have a much more diverse range of proteins, that they have a larger abundance of proteins within. Because as you can see, the green bars are much higher in the number of proteins associated with them than our blue samples, which are the winter ones. This indicates then that the head samples in the summer have a much broader range of proteins and a much more vast range of proteins expressed that is not observed within our winter workers. And this is something that we're kind of drawing a parallel with, is that we see that the winter workers seem to have a reduced proteome expression, and with that then it could be a means of reserving energy, increasing longevity. And one thing I would touch on now in another few slides is that the winter workers do have quite a large amount of carbohydrate metabolism proteins. And there are some specific pathways within that I'll be discussing in a moment that are indicating that they have dual functionality, which will help with energy metabolism, or energy generation, and also help with the immune system and reducing oxidative stress within our cells. And when we look at the volcano plot for the abdomens, what's really interesting here is that the abdominal volcano plot comparison, the results of that mirror what we're seeing in our head samples. So again, we see quite a large fold change occurring within our summer samples. And when you look at the protein distribution here, a lot more of the proteins are shifted to the inside of the volcano plot, meaning that there isn't a huge amount of protein abundance occurring within our winter samples, whereas there's a much more diverse range occurring within our summer samples. And again, we see a huge increase in cuticular proteins within our summer samples, meaning that there is not a huge amount of protein abundance or protein expression occurring for cuticular proteins within the winter. And once again, we do see an increase in xenobiotic metabolism proteins within our winter bees that's not observed within our summer bees. So it's really interesting to see that the abdomens and the heads both are following the same trend and the same pattern of a reduced proteome expression in the winter and quite a vast range of protein expression in the summer at a much higher abundance as well. So there seems to be like a two-pronged train of thought here. So again, we take our proteins that are statistically significant. So the proteins, I'll jump back, the proteins that are marked in blue that are outside those dashed lines, we're calling statistically significant. And we place them in to do some gene ontology analysis again. Once again, we do see a vast range of proteins in green, meaning that they're highly expressed in our summer samples, increased in abundance in various pathways that is not seen in the winter samples. Apart from, like I mentioned a slide ago or 2 slides ago, was this carbohydrate metabolism, which has a much greater amount of proteins observed in it in the winter than in the summer. What we see specifically is that within the broad spectrum of carbohydrate metabolism, proteins associated with galactose energy metabolism and ascorbate and pentose metabolism seem to be increased within our winter samples. And these 3 pathways have roles in energy metabolism, as well as immune functions, regulatory functions, and antioxidant pathways as well. So it looks like a reduced pathway, reduced protein expression within the winter, but specific proteins seem to be expressed with dual functionality in order to aid with longevity and also aid with energy production, which is needed for thermoregulation within the hive, as well as making it through the winter. So it was really interesting to see the shifts in protein expression here. And as a beekeeper, I thought it would be really interesting to have a look at the venom sac proteome of our samples, because you do tend to get stung quite a lot. And what I found that was very interesting is the large abundance of bee toxin proteins or bee toxin associated proteins within our summer bees that is not observed really at all within our winter samples. So it does give some indication that the summer samples or the summer bees have a larger abundance of bee venom associated proteins in comparison to our winter bees. And as well as that, they do have an increased abundance in proteins associated with oxidative phosphorylation, which could be attributed to the fact of an increased amount in bee venom associated proteins could bring its own risk factor. So to help with potential contamination of themselves with these proteins, they will increase the oxidative phosphorylation proteins to help combat any issues that could occur with having such a large abundance of bee toxin proteins. And what we see over here on the right-hand side is, unlike the abdomens and the head samples, there seems to be a range of proteins increased here, but none of which are of a specific group. So we do not see specifically a large amount of xenobiotic metabolism proteins. What was really interesting about our winter venom sac proteins was there seemed to be quite a reduction in the amount of proteins expressed here, both in the relative fold change that we see, but the number and the variation of the proteins. So the venom sac of the winter bees seems to be much greatly reduced in comparison to the summer ones. And not only did the summer bees have a large amount of proteins associated with them, 3 of which were actually exclusively expressed in the summer samples, so therefore were not actually detected within our winter samples. And again, the fold changes that we see within our summer venom sac proteins are much greater than what you see in the winter, meaning that, again, the summer bees have a really high amount of these proteins expressed, and that is all relative. So it means that the winter bees actually have a very low level of these proteins that has been detected with this proteomic analysis. And what we're thinking of this shift within the bee venom associated proteins in the summer in comparison to the winter is that the summer bees have a much broader spectrum of invaders that they would have to deal with in comparison to the winter bees. So this could be a potential reason why there is abundance of these bee toxin proteins produced in the summer and that are absent from the winter. And again, it could also be linked to everything is energetically costly within the cell. So by reducing the amount of bee venom proteins within the winter could also aid with longevity and reducing oxidative stress or cellular pressures within our bees in the winter. So to start wrapping things up, the very first thing that I was mentioning about the emergence of the winter bees, when discussing with beekeepers about when do our winter bees emerge or the Ditchinus caste emerge, the thought process is at the moment that these winter bees start coming into fruition in late autumn, and that they start as newly emerged workers within the hive like the summer bees and will transition to earlies and lates, and that when it's coming to that autumn time will actually just remain in the hive and will transition into this Ditchinus caste, in contrast to those that in summer will transition from emerged, early, late, and foragers. But what the hierarchical clustering has shown is that what if it's not the case of this linear transition from newly emerged workers in the autumn months into the winter caste, but instead in September and October, the newly emerged bees are actually emerging proteomically different from the summer bees, indicating that the Ditchinus caste actually starts when they emerge and isn't something of a transition. And that the bees that reside within the hive in September and October are actually those that have emerged as winter bees and are those that reside, instead of those that emerge as possibly summer bees that would then transition within their lifetime. So this is something that we want to investigate further. And at the moment, our thought is, what if our newly emerged bees in September and October are possibly fed a different food substance that is maybe changing the protein expression, enabling them to have a greater tolerance to oxidative stress, and therefore maybe aiding or contributing to longevity. And the discussion between the winter and summer bees, some of the big take-home messages that I've found while doing this investigation is that our summer bees have a high level of protein expression. So the proteins that are found within the bees are actually expressed in quite a high abundance in comparison to our winter bees. So you can detect the proteins at a much higher level. And not only that are they expressed at a high level, there's actually quite a wide range or a diverse range of proteins expressed within the summer bees, but there's only a conservative amount of proteins expressed within our winter bees. But in contrast to this, the proteins that are expressed in a high amount in our winter bees seem to be quite specific, have dual functionality, and therefore could help contribute to energy metabolism, energy production of the winter bees, aiding in thermoregulation, but also contributing to longevity, allowing them to live for such a long time within the hive. So these are some of the big points that we're investigating and continuing to develop, and it's been really exciting looking at it. So what's really intriguing about this whole thing is that at the moment, the proteomics that we've conducted seems to indicate that the bees that are emerging within September and October seem to be almost significantly different from those in July and August in terms of their proteome expression, and whether this is to do with the external environment or if it's to do with food or maybe behaviors in the hive that we're unaware of at the moment, is something that we're really intrigued to continue the research and follow up on. And that the bees, the winter bees that are surviving through the winter months in these temperate climates seem to have a really specific amount of proteins that they have increased in abundance, in contrast to the summer bees, which have a myriad of proteins that they're using to complete and be able to do their roles so successfully within the hive. So it's been really interesting to see this contrast in protein expression within our workers. So I'd like to thank everybody on the team, both Clarence Ireland and Maynooth University, for allowing me and helping me to do this research, Professor Kevin Kavanagh for being such a fantastic support, my co-supervisor Mary Coffey, and a special thanks to Danny Kyo, who was a beekeeper down in Carlow, and he's taught me so much. It's been so wonderful to work with you both. And thank you all very much for listening, and I'd be happy to take some questions.
Thank you very much, Rachel. As Rachel has said, we were happy to take any questions that you might have. If I could kick off some questions, Rachel, in your work, you show that the bee that emerges in September and October is quite distinct to the bees that emerge in the summer. So what do you think is the trigger for this?
I think it could be a few triggers. I'm curious to see about the weather around the hives at that time. But one of the main things that I've been thinking about and beekeepers have been talking to me about is the nutrient availability, the food source, and what the bees are actually being fed in their egg and larvae stage that's causing this distinction between our summer newly emerged bees and our winter newly emerged bees. So it's something I'd really like to investigate and look into some more.
Yeah.
And
there is a question about that, or related to that, Kevin, if I may. Adrienne van Sandvik says, if I understand correctly, the tested bees born in the incubator are born in a state of hopeless queenlessness. Will this not influence the test results? So this is in the realm of, you know, what influences.
That's interesting. I will say that the newly emerged bees are kept within the incubator, I believe, for a day at most. So it really is, they all seem to emerge quite quickly, are marked on that same day and returned to the hive. So it's very like labor intensive for us to do, but we're very aware of the impact of sampling with the hive as well, and the impact of the stress and being without the queen. So we did try and work as fluidly as possible, but we knew that this was something we'd have to do in order to get the exact age of the bees for future sampling. So that would be an interesting aspect to think about as well.
So a lot of different aspects like surrounding that you can still test on. So Kevin, can go on.
So Rachel, with your hat, putting on your hat as a beekeeper rather than as research scientist, how do these findings, or how might these findings influence how beekeepers might treat hives approaching the winter?
It's really interesting. It's been something I've kind of been wondering myself as well. And especially coming up to this winter, I have been looking at how the weather as well as affecting this transition of the bees and when are the winter bees emerging. And the biggest thing that I've been paying attention to with the hives that I've been dealing with is monitoring them to see what their disease levels are like, their food sources are like, because it's around this time. Before this, obviously it's December, but in August, September and October, we were really paying attention to the quality of life that the bees had, how stable they were in the hive, because I understood now that these are the bees that are going to be surviving through the winter. So we just thought it was really important to keep a check on them, and do they need treatment or do they need additional food? And just monitoring them is definitely key now, and I would advise beekeepers to do the same, I think.
And it might inform when winter treatments are applied to make sure the winter bee is there rather than too early.
Yeah, quite possibly. Absolutely. That would be something that is really important to understand. You know, you want to treat your hives to make sure that they live throughout the winter. So identifying when is the best time with regards to when the winter bees are emerging, the ones you want to protect, will be really influential or really important.
Yeah, what is the trigger, right? Is it the sun, the light?
So this is it. It's really interesting because we've had quite a warm spell here within the September and October months. So it's been really interesting to see that the bees are still, you know, going in and out of the hive. So whether it's the light, whether it's the warmth, whether it's the change in the day cycle, I'd love to be a bee and ask them, if I'm completely honest.
So we have a question from Grace McCormack. Lovely talk, Rachel. What happens the winter bees in spring, do you think? I also had this question. Yeah, how is the transition? Is there another switch in feeding protein expression?
Yep, so we have actually sampled from December to the following March. And in the March, you do start to see these bees transitioning back to that like nursing stage, because obviously these are the only bees within the hive when the queen starts to lay. So they have to resume nursing duties. And with the presence of brood, it seems to trigger like an almost internal clock, or like reset their internal like lifespan or life stages. So it's really interesting to see this dynamic switching of this age polyethysm mixed with this winter-summer cast that's occurring. So it does look like the winter bees will switch their behavior back to nursing duties, and then one transitions through the hive to go to the lates and then the foragers, and will subsequently die quite quickly after being foragers.
One additional question, maybe I didn't get. It was very fast, a lot of protein.
Oh, I'm so sorry.
No, it's okay. It's just maybe I didn't get it and you explained. So do the winter bees forage?
The winter bees will end up foraging, yes.
End up foraging, okay.
Yeah.
For clarifying that. Kevin,
That's
go
okay.
on. Kevin, would you, do you?
Um,
Will I read the next question?
sorry, okay.
Vincent Boire.
Yeah. So there's a question here about the difference between Northern European bees and bees around the Mediterranean. Do you think what's going on at the Southern Europe? Do you think?
Really interesting. I would be curious to see if you took bees from the Mediterranean region and brought them to temperate climates, would they adapt and like develop this ditchiness cast, this winter cast that allows them to live throughout the winter, sorry. And then vice versa, what if you took our temperate bees, Apis mellifera, and brought them to the Mediterranean to see, would it be a quick shift in roles that would allow them to survive, or would they actually die because they didn't know how to deal with it? They'd go into automatic shutdown mode and for the winter, for our temperate bees to the Mediterranean, and then would the Mediterranean bees really struggle with the climate? So it's something I'm unsure if it's as dynamic and quick in terms of climate changes, like climate changes in temperate and Mediterranean. That would be quite interesting to investigate. I'm not sure it would be nice for the bees, but yeah.
It would be also interesting, you know, when we say Ligustica, they don't overwinter so well, they just keep on breeding and so on. If, you know, they don't have this shift to winter bees or not as much, we're comparing, you know, find probably differences within, like, among different subspecies there. That would
absolutely. And when you look at the, oh, sorry, go ahead.
No, it's good.
Isn't it at the moment when you've got the issue with the buckfast bees coming to Ireland, that even between England and Ireland, the bees are quite different in dealing with the climate, because Irish climate is wetter than England. So having to adapt to that already in your two islands that are quite close together. So even at that, the bees tend to be quite specific to their region and to their climate, which is quite interesting, wondering how quickly they might adapt.
There's one question which relates to the microbiota in winter versus summer bees. Have you come across anything about that in your studies?
I haven't. I have done some reading on the microbiota and how important it will be for the pollen and the nutrient availability. And I know there is current research coming through with it, but I myself haven't looked into it in a great amount of detail. I know colleagues of mine are doing it on bumblebees at the moment, and they're finding it extremely helpful with their research as well. So it would be something that could be explored in the future.
Mm-hmm. And I just see one other question which relates to morphological differences between winter and summer. Is there any information on that? Do we know the different morphologies?
I cannot say that I do know. All I know is that when a bee emerges, it's much fluffier than when they're fully mature adults. So that's the only physical morphological difference I can spot. And with the winter and the summer, not that I'm aware of, there could easily be. This would be my second winter coming now, so I would definitely keep an eye out for that.
Okay.
Yeah. So there is one from Andrea. Hi, Andrea. Good to have you here. Have you looked to see how the protein profiles change in the winter bees change as they progress over the winter?
Yes, this is something that I do have, and I'm just waiting to kind of sink my teeth into it to see exactly what's happening, because it'd be really interesting to see if specifically the carbohydrate metabolism proteins are possibly decrease as you're going through the winter, with food maybe becoming a little bit more diminished, or if it's increasing or staying the same. So it is something I have the data for. I just need to investigate. So I'm really excited to do that.
Yes.
And we've used Apis mellifera in our work. One question asked, does this apply to all subspecies? We can't give a definitive answer, but we've looked at Apis mellifera. So that was our work.
It's hard not to work with dark bees in Ireland.
Oh yeah, there you are.
Fortunately. Okay. There were no benchmark with other bee breeds as part of your work. I don't understand quite a question. There's another one from Vincent Douard.
Yeah. So we haven't benchmarked with other bee breeds. It's not part of our work. And then the other question: what trigger do you think is operating for the bees to shift to become winter bees and also to forage with the presence of brood in early spring and nursery tasks? So I suppose this addresses the question: at the end of the winter season, the winter bees then have a number of tasks to do, and is there a trigger that kicks them into action to begin nursing?
Mm-hmm.
Yeah, it's intriguing because I wonder if that's the same trigger that it kicks them into becoming winter bees. So is it the queen that is reducing laying, which is almost telling the bees, now is the time, get into your winter phase, or do they emerge like that? So that's what we're thinking there. But kicking it into the summer, is it the warmth that's causing the queen to start doing? Does she have some sort of internal clock because she does have a much greater lifespan than the workers? So it's really interesting to explore. Is it the brood that's doing it? Is it an external stimulant? Gabrielle, you mentioned, is it to do with the lighting that's happening because of the additional light sensory glands that they have on the top of their head, or the light sensory organs that they have on the top of the head? So is it something to do along those lines that's causing these triggers? And it's definitely a question that is buzzing in my brain, really, to try and figure out what's happening.
I wonder, is it the commencement of egg laying that's the trigger? That as the queen is beginning to lay egg and lay up the brood, that's actually a hormonal trigger, then kicks them into becoming nurse bees.
Mm-hmm.
Yeah, is it some sort of scent that's picked up by the odorant binding proteins that's causing, a cascading trigger that's happening then to jump them back into the production of the major jelly proteins? The fact that they're at such low levels within the winter bees, and those are the proteins used within our bee milk. So is it something linked with a flactory, a smell within the hive that's causing them to be able to increase these particular group of proteins to help with brood rearing then afterwards?
There's a follow-on question to that, and from Vincent, and it says, do we think, are there social triggers or pheromones, a chemical trigger? But we don't know at this point. There's one question from Jonathan on, I read it out: I was interested in the result you mentioned about P450s. I know P450s are important in detoxification. I think you said they were more prevalent later in the year. Can you tell us more about why there might be increased expression of P450s in the late bees?
That's a great question. It's something I found really interesting as to why that group of proteins is increased in such a substantial amount within our winter workers. And I do believe it's a mechanism for them to aid themselves with longevity. So detoxifying themselves. The P450s also do have roles in the antioxidant proteins, and there are other xenobiotic metabolism proteins that work in conjunction with our P450s that will aid in that longevity. Specifically why, it seems to be almost like a defense mechanism, I suppose, of the bees. So instead of proactively, increasing their oxidative phosphorylation proteins, which can themselves generate reactive oxygen species, they've kind of decreased the OXPHOS proteins and have an increased abundance in these cytochrome P450s, almost as a defense mechanism for cellular stress instead of maybe an offense mechanism. I play football, so that's like how I'm thinking about it. But that's kind of my train of thought at the moment as to why these P450s are increased within our winter bees. It's almost like a safeguarding for themselves for this cellular stress that they could be coming across. And I also wonder, is it due to the fact that treatments are usually applied later in the year? So has it been something that has been developed? And that's something I'm looking at because I'm doing research within these lines of my project on how various antiviral treatments are affecting our bees as well. So it's really interesting to kind of investigate that in parallel with this work.
Excellent. There's a parallel question to that question, which is about the immune protein expression in summer and winter bees. So if we look at in a global sense, what can we say about immune response or immune proteins in winter versus summer?
So in the immune bees, we did see defensin, which is a known immune response protein, but that wasn't observed within our winter bees, which I thought was quite unusual because I was expecting more immune proteins to be present within our winter bees. But again, it looks like this quite reserved proteome is occurring within our winter bees, and instead they're selectively choosing some proteins that have dual functionality, like I mentioned with the ascorbate, the galactose, and the pentose metabolism, that have dual functionality with energy as well as antioxidant and immune proteins. So it seems like the winter bees don't produce these immune proteins, these antioxidant proteins, apart from the P450s, unless it's needed, in a sense that if you produce the proteins, it costs energy. The bees in winter have a finite resource of energy that they're going to be able to use. So they're quite intelligent with the proteins that they're expressing in order to be able to reserve energy levels with themselves and then subsequently within the hive. So I do believe that's what we're seeing there. And in the research I've been doing, in collaboration with all of this, it seems in general, when I'm looking at our bees, even with age polyethism, that the immune proteins aren't really expressed unless there's a stressor that's been given to them. So it does seem they seem to be a little bit more on the fence when expressing these immune proteins.
Okay, there's a very pertinent question, and it says, we all want to live longer. Is there any extrapolation to other taxa? So can humans learn anything from bees?
I'm not quite sure. The train of thought at the moment is the royal jelly, because that's the thing that the queen feeds on the most, which has vitellogenin. And I know I didn't mention it here, but the vitellogenin is seen as statistically significant, differentially abundant, so significant for the winter bees. So it is one of the proteins that I find in higher abundance in our winter bees in comparison to the summer. So maybe that's the key. But I did get a chance this year to try royal jelly, and it doesn't taste nice, so I wouldn't recommend it.
Okay, and then you might remember the setup for the trials. One question asks, were they treated for Varroa?
The bees were treated for Varroa this year because there is a lot of hives in this apiary that we were dealing with, and we were very conscious that there was some other trials going along in collaboration. So we wanted to try and keep them as controlled as possible, as well as assuring their survival throughout the year, because drifting effect we have observed. We did observe, actually, quite interestingly, with our age polyethism study. So we needed to make sure our hives were safeguarded from Varroa as much as possible, because what was quite interesting, we had 3 hives in the apiary, and those that were marked were actually found in other hives as we were continuing hive inspections throughout the apiary. So that we did actually visibly see drifting occurring throughout the hive. So again, if I could be a bee researcher, definitely I would look at drifting within apiaries.
Okay.
These are great questions.
Andrew has a question. Okay, let's see here. Okay, so one question: is there a fat switch mechanism that hibernating mammals show? Is that known in bees? Do they build up adipose reserves and that triggers them? But I don't think so.
I'm not aware of that happening. I do know there are changes to particular glands. The hypophalangeal glands within the head shrink in our winter bees, and the fat body composition also changes. But with fat reserves, I'm unsure of that, because I think insects being a completely different caliber to mammals, I'm sure they would have come up with some different adaptation to do that.
There's a question from Jonathan relating to why did Clarins fund this project? Well, Clarins, as you know, they're an internationally famous cosmetic and skincare company, and many of their products are derived from plants, and particularly from flowering plants. And they wanted to contribute to the work to preserve and protect the bees because of their essential role in pollination. And so they very generously supported this project as a way to give back to the bees for all the work they've done for them over the last 50 years. So that's why they did it.
And it's a question from Mary and not from John. They specify because they watch together. She probably knows Clarins.
It's been lovely to work with them. They're fantastic support, and they are really invested in looking after the bees and being sustainable with their beauty range and everything like that. And they've actually implemented bee boxes and bee friendly boxes around their location in Dún Laoghaire. So it's been a pleasure to work with them.
Well, that's great. We're going to take this into our fundraising department as well.
You can tell them I sent you.
Somebody has a question about bees drifting, and that's well known. And that, but they're asking how far do they drift? You know.
I don't. I'd love to know. I do wonder, because they have such a wide radius that they can go outside of their hive, that maybe if there's one at the perimeter that they see a lot of activity. I don't know. It'd be interesting to see. And like I said, if I had access to, maybe I'd do it with my own apiary, mark loads of bees and then do some photographing.
I think there has been quite some work been done on that.
Okay. Yeah.
Like Alder Day, and I think we can find Matties. We can find some if you like.
Yeah, do.
Okay. A question from Maggie. We're not aware if any other work has been done with similar work has been done with other species. And then it's from Vincent. It says, it's said that the winter bees are choked at lower temperature than summer bees. So is there any information on their thermo resist incubated? Okay, I always got it. Sorry. It said that It said that winter bees are incubated at lower temperatures than summer bees, presumably in the brood. Do you think that is what's going on?
Possibly. Interestingly, I did see heat shock proteins increased in the summer bees. So whether that's contrary to that, I'm unsure. Again, that would be one of the factors. I'm constantly questioning this answer of what is it that's triggering this difference in our September and October emerged bees in comparison to those in summer. So you're giving me loads of ideas as to what it could possibly be. And it's amazing to think the amount of influences that could be happening with our bees to cause them to develop in this way. But this is obviously something that hasn't just happened this year, the year I was doing it. This is something they finely tuned. So it's something that's consistent, and whether it is a lower incubation temperature.
Yeah.
Interesting.
So heat shock proteins, they might also develop because it's hot. Like
exactly.
we develop, like we express heat shock proteins in the sauna, right? That's what the good thing about sauna.
They do. Yeah, there could be that could be what we're missing,
Not necessarily in the brood, right? So that it can develop during foraging, right? Yeah, I don't know. Oh, you have so much more to do, Rach.
Loads.
Great.
I have one more question about the venom sac. That was interesting, as being highly allergic against bees. So my question is, can I go and do some beekeeping in the winter safely?
I'm not going to say yes.
I'm sorry.
Oh, you're okay. But don't get me wrong, if you go and annoy a winter colony, if you have to feed them, they can still sting you, and it does still itch. So I wouldn't recommend it. But I just found it so interesting that there was such a huge difference between the protein expression between the two.
There was also the allergenic protein allergy. I think there is a whole range of allergenic proteins. This might not be the only one, but it gave me kind of hope. Maybe we can GMO bees to not express allergenic venom.
Quite possibly, yeah. You got a few of them. You could just maybe knock them out with some of your gene research.
So that might be why beekeepers say the stings are feeling different in the spring. You always assume it's because they didn't get stung for a while, but it might also be due to the differences in venom. Right.
I've heard that too. Maybe it's because your immune system isn't primed for being stung, and maybe it's the two combined.
So that was very interesting. Well, Rachel, still a lot to do. So Mary says great, thanks. For the Clarence. She's probably writing a draft right now to send to Clarence as well. That was very interesting, Rachel. So what will your future work look like? And also
so
to Kevin, yeah.
I will be finishing up soon, so I still have all the research, all the beekeeping work has been done. I have a lot of data analysis to complete with the treatment trials, one of which has actually just been accepted for publication, so we're delighted with that. And then we've got H. polyethysem to investigate, as well as the diseases, and then the winter comparison. So I will be on my computer for the next few months anyway, compiling all this data.
And to you, Kevin, do you plan on continuing from Rachel's PhD, continuing this work with the winter?
Well, absolutely, I would love to, but as in all these situations, we need to get funding to do that. So we will pursue a number of avenues to see if we can get funding to continue this work. Yeah.
Well, we sure are looking forward for future results. So I understand we might hear some more at the next conference, right?
Excellent. Excellent.
Okay. Thank you very much.
Thank you, everybody.
Thank you.
And to you all, thank you for attending. Again, also for all the interesting questions. That's always very nice. And I would like to point out the next talk next week. It will be Marina Meixner on the Your Best study for varroa-tolerant bees across Europe. That will be very interesting too, and I hope you will all attend again. So we give you some minutes to exit the conference and hope to see you soon. Thanks. Bye-bye.
Thank you.